welcome to Biochemistry. Biochemistry, as you know, is a YouTube channel dedicated to clear fundamentals of biosciences. In these videos, we are going to discuss about carbohydrate derivatives. Several monosaccharides undergo modification, and these modified products becomes very useful and they act as very versatile molecule in signal transduction and also in metabolism. We will look at various modified or derivatives of carbohydrates, like we will look at phosphate derivatives, we will look at amino sugars, we will look at phosphate sugars, we will also look at the acidic forms. We will also look at deoxy forms. So these are some of the sugar derivatives we are going to address today. Let us begin by looking at amino sugars. There are the most important amino sugars are glucosamine and galactosamine. Amino sugars, the most important them, glucosamine and galactosamine. From these two derivatives, many other forms have been made with a slight variation within the structure. More than 60 types of amino sugars have been identified. What is happening in the case of amino sugar is one of the hydroxyl group is replaced by an amino group. So let us write the structure of glucosamine. So this is glucose. This is glucose. If the amination happens at the second position, this is position 1, 2, this is 3, this is 4, 5 and 6. At the second position, an amino group is added. So therefore, this becomes glucosamine. Glucosamine is found in mucopolysaccharides and it is also found in blood group substances. So this glucosamine has varieties of functions to perform. We will also take up along with the glucosamine N-acetyl glucosamine, another derivative of glucosamine. What is the modification happening within the structure of N-acetyl glucosamine? An acetyl group is added. So therefore, this is COCH3. This is N-acetyl glucosamine. So this N-acetyl glucosamine is generally added to proteins. We know that where a serine residue is present, so a serine residue will have a hydroxyl group. A serine residue will have a hydroxyl group. To this hydroxyl group, N-acetyl glucosamine or N-glucosamine is added. So therefore, by loss of a molecule of water, it forms an O-glycosidic linkage. O-glycosylation of protein is indication of stress. When there is hyperglycemia, that is increased sugar levels in the body, because of the O-glycosylation, body becomes resistant to insulin. So resist insulin response. And it is also been associated with cognitive function. Therefore, glycosylation of protein seems to be decreasing during as we become older. So it affects cognitive functions like memory, it's affecting. And the glucosamine is also used as a kind of a supplement, especially in rheumatoid arthritis. Because N-acetyl glucosamine and glucosamine are part of mucopolysaccharide. Another carbohydrate derivative is muramic acid. So this is the structure of muramic acid. Look at carbon number 3. There is a modification in the structure of muramic acid. Another variation that happens to muramic acid is N-acetyl. N-acetyl is added to muramic acid. So that can be done by adding an acetyl group here. So this will be the structure of N-acetyl muramic acid, which is found in the peptidoglycan of gram-positive bacteria. Another amino sugar is the derivative of galactosamines. The structure of galactosamine will be like this: 6-carbon sugar, this will be galactose, and this is galactosamine. Galactosamine and as well as N-acetyl galactosamine, that is with the variation in COCH3. This is N-acetyl galactosamine. These molecules are found especially in certain hormones like follicle stimulating hormone as well as luteinizing hormone. Remember that these molecules are hepatotoxic in large concentration. We should also keep in mind that these amino sugars do not give ozones. After looking at all these amino sugars, we are going to look at another important amino sugar, which is sialic acid. Sialic acid is also known as N-acetyl muraminic acid. So sialic acid, another name for sialic acid is N-acetyl muraminic acid. This is a very important amino sugar. This amino sugar is a 9-carbon sugar, and it was originally found in the saliva. So therefore this name came sialic acid, and it was discovered in 1952 by a scientist whose name is Gunnar Blix. A Swedish scientist identified sialic acid from saliva. We know that sialic acid is abundantly found in bacteria. It is found in archaea. It is found in all animal tissues, especially in the glycoproteins, glycolipids, in the brain. Sialic acid is abundantly found. This is found abundantly in all vertebrates as well as in certain higher invertebrates. Sialic acid is found. It is not found in plant species. There are more than 50 different types of sialic acid identified so far. And sialic acid, because it is present in brain, we know that sialic acid is involved in cognitive functions like learning, like memory. Sialic acid is playing a role. We will look at the structure of sialic acid. When we look at the structure of sialic acid, we realize that it has got too many negative charges on its surface. And these negative charges makes especially the RBC coated with the sialic acid, makes the RBC repel against one another, as a result of which RBCs will not be clumping together. We know that clumping of RBC is known as Rolex formation. It can be prevented because of the negative charge that is present on the sialic acid. And sialic acid is also associated with the altering in the half-life of a protein, especially under pathological conditions. We are going to look at the structure of sialic acid. Sialic acid is basically made from two molecules. One is pyruvate that combines with the mannose. Mannose, we will be modifying the structure of mannose. we will make it mannose-amine. We will do a condensation, an aldol condensation reaction with the pyruvate, and we will make a 9-carbon sialic acid. As we know, this is the structure of pyruvate, a 3-carbon compound. This is the structure of mannose, and in the monomeric unit will be mannose-amine. In the second position, it is NH2, so therefore this is mannose-amine. So there is a condensation reaction happening between these carbon atoms, as a result of which you have this compound, which is nothing but sialic acid. So therefore, let us number the carbon atom. This is number 1, 2, 3, 4, 5, 6, 7, 8 and 9 carbon atoms. Now in this structure, in the second carbon atom forms a glycosidic bond with the sixth carbon atom. So therefore, it forms a ring structure as follows. So this will be carbon number 1, carbon number 1. This is carbon number 2, carbon number 3, 4, 5, 6. 6 and the second carbon atom, a bond is formed. And the remaining number 7, 8 and 9 will be part of the structure. This is how the ring structure of N-acetyl neuraminic acid will be shown. Neuraminic acid is a very interesting molecule. There are two forms of neuraminic acid. One is N-acetyl neuraminic acid, and the second form is N-glycolyl neuraminic acid. Human beings, N-acetyl neuraminic acid is present. This will be NHCOCH3, and in this case it will be NHCOCH2OH. This is the difference. Now, many times what happens is with the consumption of non-vegetarian food, milk, poultry, all these can add N-glycolyl neuraminic acid into our system metabolically. So that once it is added, what happens is certain bacteria, they will use that as a kind of binding site to cause infection in our body. Several examples like influenza, Helicobacter pylori, Plasmodium falciparum, all these are known to attack or infect cells by doing this kind of molecular mimicry. So this is a brief account about a neuraminic acid which is found in several parts of our body, and we looked at what are the characteristics, what are the functions of neuraminic acid has in living system.